However, in synthetic biology, there is a serious criticism to this concept because of the following. So let's take the concept to an extreme, that you take one thing, and then that thing gets connected to another thing, and then to another, and then it goes a little better, and this and that. Okay, so if you take that concept to an extreme, you end up in a situation that has been kind of converted into a cartoon by this gentleman called Rube Goldberg. That is, I think he was a Belgian cartoon drawer, that was as follows. So imagine, for instance, that you want to develop a function to wake up this guy in the morning, okay? Then say, okay, no problem, you know. So we have this bird here that will get a worm, and then the worm will cut a thread here, and that will trigger a pistol, and the pistol will trigger a bullet that will explode a balloon, and then there's a brick here, the brick will go into a sprayer, the spray will spray some water to this sponge, and then the sponge will be heavier, and then you will have this coming up, and then you have this ball on the face of this guy, and then we'll have this type of thing. So, I mean, this is something that in biology we observe all the time, and we don't give to that any importance, you know. So we see sometimes a tremendous complexity on things, and we take for granted that, well, okay, so it's just evolutionary history. Okay, does it make any sense? You know, is that something that we can take for granted? Wouldn't it be better to have a kind of alarm clock, or just to have this bird hitting the face of this guy directly? I mean, this type of, can we make biological processes easier to make them work? This is one of the questions of synthetic biology that are interesting. Okay, so the answer to that is that evolution will not tell you anything about the way things work. They can tell you, evolution will tell you where things come from, but nothing at all about how they work. And this is because of the following. There's this phenomenon in evolutionary biology that is called exaptation. So exaptation means that one function, one object, one biological, say, asset, emerges in evolution to fulfill a given function. But then it happens that at the end, the original function is left behind altogether, and then the same object acquires a completely different function that is alien to what happened before. So the typical example are feathers in dinosaurs. So feathers were, say, invented evolutionarily to provide thermal insulation to dinosaurs. However, I mean, the big success of feathers was provide a mechanism for flight, okay? So given that, if you say, well, I have a feather here, then evolutionarily goes into a thermal isolation function, and then will that predict that this thermal isolation will be converted later into an asset for flight? No, not at all. So we have to be careful on using evolutionary histories of different genes and different functions as the basis to determine the final role. And this is something that, again, synthetic biology keeps in mind very, very strongly. And therefore, there is this question on, given all these circumstances, how to describe, how to deconstruct, how to reconstruct biological complexity, because we are all aware that biological systems are complex, and it's not just an intuition. Now we can measure complexity, analyze each of the members of a complex system, the interactions between dimensions, and we do see that there are many, many components that interact in many complex ways. So can we, I mean, how can we do that? Okay, so we have to, again, make a kind of conceptual consideration that goes as follows. There's this concept that has emerged mostly in the last 50 years or so, that is the so-called postmodern approach, and this concept pioneered by this philosopher called Gianni Vattimo and others, that propose strongly that the ultimate, say, nature of things is difficult to grasp. So instead of having facts, what we have is or are interpretations, okay? So all we can do when you face complexity is to interpret the complexity, but forget about, or at least put aside for now, understanding the complexity. So, then we have to find the right conceptual frame to tackle complexity. So, and we can adopt what kind of conceptual frame is the best to describe biological complexity. So there are three, say, frames or various frames that you can adopt. And just remember that, that when we look at things, we always pass these things through some kind of filter, and the filter is this conceptual frame that we have already adopted, either explicitly or not, to understand what is in front of us. I'm sorry that this might be a little theoretical, but we will run through that very, very quickly. Okay, so the bottom line is that all languages for describing reality are metaphoric, okay? So metaphoric means that they are an interpretation of the reality. So they are not the reality, but an interpretation of the reality. Then that means that for understanding complex systems, like biological systems, we need to adopt adequate metaphors, adequate conceptual frames. And third, and this is perhaps the most important, no descriptive language is neutral. They all have an agenda, okay? So you say, well, this is not true. I mean, you have mathematics, 1 + 1 is 2. I mean, there's no conceptual. Okay, is that completely true? I mean, even mathematics has behind some type of conceptual, say, frame that is not identical to other conceptual frames. So mathematics, you can say, well, is one attempt. The agenda of mathematics is trying to understand nature and to quantify nature and to ultimately dominate nature through this kind of understanding. So, well, what type of language then would be adequate to take in biology to handle this complexity, and what agenda we have to do that? Okay, so here comes synthetic biology. That is the first kind of slide where this story is, or this concept is explicitly mentioned. So, synthetic biology is nothing more and nothing else than understanding biological systems using engineering as the conceptual frame to tackle what is going on in them, okay? So synthetic biology is looking at life systems using engineering as the metaphor or interpretive frame for making sense of what we have in front of us. So, well, this kind of, say, vision is logical. And I insist that, depending on the interpretive frame that you adopt, you can see in the same object completely different things, okay? So if you show this picture to a microbiologist, someone that is trained in microbiology, and you ask, what do you see? What would you say? Well, I see bacteria. I see cells. They have these rod shapes that are typical of bacteria. Maybe they are interacting and transferring DNA. They have some, say, structure here. Some have the surfaces. There may be some proteins on the surface. I mean, this is the typical view that microbiologists have of bacteria. However, if you show the same thing to a molecular biologist, they will see other things. The molecular biologist will see molecules all the way around. This is a cartoon of one of the first mappings of this bacterium that is called Mycoplasma. That is, in fact, the first bacterium that is nearly completely understood. And every protein, every gene, every RNA has been mapped already, and we know exactly where every molecule is in this bacterium. What you have here is the vision of a molecular biologist of a bacterium, right? So in this case, a molecular biologist would not be interested to see the shape or to see the interactions, would be interested to see that there are proteins, that there are ribosomes, that there are chaperones, there are interactions, RNA polymerase, and all the rest of it. So it would be the vision of a molecular biologist. But if you show a bacterium to a computer scientist, what do you see there? Okay? So a computer scientist would see a machine that is able to process information. He would not care, or she would not care about the shape, about the molecules, about all these things. All you want to know if you are a computer scientist interested in biology is that you have a information processing device that is able to take inputs, process them, and convert them into outputs, following a given logic. And in this case, you have here logic gates. We will return to that a bit later. And you know, you can ignore completely whether these processes are mediated by enzymes, by RNA polymer. It doesn't matter. All you need to know is that you have something here that you can consider like a device that can take information in the form of inputs and release outputs in the shape of activities or in the shape of different behaviors. Now, if you show bacteria to a biochemist, okay, what do you see in a bacterium? You see this. I mean, you see that, in fact, you see much more complex than this. You see a whole bunch of metabolic pathways. So you see reactions, and you have, if you have been through biochemistry courses and lectures, well, you have all types of interactions here, the Krebs cycle, all the rest of it. So, and in that case, the biochemist will not care about the structure of the RNA polymerase. He will not care about the shape of the cells. All they will be concerned about will be about the chemical network, the biochemical network that you have there. If you show a bacterium to a chemist, not to a biochemist, but to a chemist, what do you see in a bacterium? Well, what you see in a bacterium is a chemical factory. And as a matter of fact, one of the most, say, instructive or more revealing metaphors in synthetic biology is this concept of cell factory. So cell factory is this idea that you have in a cell not a mixture of enzymes that run reactions everywhere at all times, but you have a quite a structure, say, system with pipes, channels, and fluxes that can run many, many different reactions in a quite organized three-dimensional fashion. So, you know, the idea is, as I mentioned, you look at the cells, but if you are a chemist, you are an engineer, you are a molecular biologist, you are a microbiologist, you are a computer scientist, you see different things. And there's nothing wrong with that, is that we are just using, or they are just using, different interpretive frames to understand what is going on. So, as a kind of corollary of what I told you, we can adopt this interpretive frame that in the language of philosophy is called hermeneutic frame. Hermeneutic is the term that indicates the interpretation. And this gentleman here, Jacques Monod, a French Nobel Prize, famous for his work in the lac operon in E. coli, he wrote this book in year 1971. It's called Chance and Necessity. And he said, and it was something that was very important for the subsequent development of molecular biology, that biological systems do not have a intention, they don't have a purpose. I mean, they appear in a kind of, say, random way, and then what we see is the subject of evolution, and there's no end, there's no finality in biological systems. However, for understanding how biological systems behave, we have to make a little assumption for a moment. Is what he called teleonomy. Let me elaborate with that. It's an important concept. So there's this word called teleology. Teleology means that biological systems have a purpose, okay? And data evidence indicates that is not the case. However, if you want to understand the way they are organized, they appear to have an objective, right? Like, you know, I have eyes, you know. So the eyes by themselves, if you look at the structure of the eyes, then they appear like if they had a kind of purpose. But in reality, if you see the mechanism and the evolutionary history behind that, then they don't have it, right? So he coined this kind of paradox between what he called teleology, that is the reality of purpose, and the teleonomy, that is the appearance of purpose. And he said, well, we can, we know that this is not happening, but for the sake of understanding, we may want to assume that things appear to have a purpose here. So by the same token, you know, in synthetic biology, there is another, interesting conceptual aspect, and it is that there is the contradiction or paradox between technology and things, how machines work, made by man and so on. And we know that biological systems are not made by someone, right? They have appeared through evolution and everything, and they work because they have a certain logic that is not human, but is produced by evolution. However, also for understanding how bacterial systems or microbial systems or biological systems work, we have to assume that they follow the same rules than the technological human-made objects and actions, okay? So in other words, that the logic, the physical chemical logic that make biological systems to work has to be identical to the logic that an engineer uses for making an object work. So objects work because they have a relational logic that is not magic, is not different from the rest of the different logics that are out there. So then the choice of synthetic biology, conceptual choice of synthetic biology, goes as follows. You can say, well, cells are design machines. This is a metaphysical statement. We are not interested for that. Or you can say, well, cells are like design machines, for a metaphor. Or, and this is what synthetic biology adopts, is that cells can be described and understood as if they were design machines, okay? So it does not mean that they are design machines, but we can understand them if we consider them as if they were design machines, simply because we assume and we propose that the logic of a cell factory in a bacterium has to be the same logic of a chemical factory. It has to have pipes, it has to have valves, it has to have some type of architectural design, and therefore, in terms of conceptual choices of synthetic biology, please remember this, that cells, biological systems, can be described and understood as if they were design machines, okay? Okay, so what are the consequences of that? Well, the consequence is that you can start with something existing, with a biological system that appears as it is. Then with all the plethora of so-called omics techniques, then you can take this apart and identify what are the different components, the interaction between them and so on. And then the typical, say, action of systems biology would go to make a model, to extract from this pool the basic, components, and then to make a model, and this model will capture part of the functioning of the bigger, more complex object, and then you can understand how this works. If this model does not respond well to the original situation, then you kind of make the model more complex and more complex and more complex. But what synthetic biology does, and please remember this slide because it's important, is that synthetic biology uses the same parts, the same components that are produced by systems biology, by genetic engineering, by analysis and so on. But instead of recreating what we had before, the parts are used for shaping a completely different thing. That is not what we had before, but it uses the components that nature has offered us to build a new thing. So this is what nature would do, and this is what we do in synthetic biology. You see, we use parts that come from biology, but we use them for doing something that has not been produced so far by biological systems as a natural product. So, synthetic biology, and this is a definition that comes from Jay Kisling, the guy that I told you before was involved in the production of this artemisinin product. So Jay Kisling at some point said, well, synthetic biology seeks to understand and design biological systems and their components to address different problems. It has tremendous benefits, and very important, as I said, that you have to understand biological systems with the same concepts, the same principles that engineers understand the functioning of the products or the devices they produce. You need to have a blueprint. You need to know, you have a plan. You need to understand the relational logic of the different things. Now, synthetic biology historically then has like three roots. So one of them is engineering, computing, modeling. I just mentioned that. On the other hand, we have to bring into the scenario molecular biology, evolutionary genomics, biotechnology, And then, and this is something that I will mention also in subsequent, talks, then there is a connection to the origin of life, artificial life, and orthogonal life. Please, remember this word, orthogonal life, because we will discuss that also at some point. And, well, if you make a mixture of them and you see the overlap, then at the core of this overlap you have synthetic biology. So synthetic biology is not something that comes out of the blue, it's something that has its origins and its precursors in all these bunch of other disciplines, some of them coming from biology, like molecular biology, but others coming from engineering, straight engineering. Historically, synthetic biology has been, the result of engineers that had no idea of biology facing a biological system. And then, as you will see later, they started to be curious how a biological system can work. Can we interpret biological systems with the frames and the tools that we have to analyze, to construct human-made devices? Can we apply that to biology? Well, this is the thing. And in fact, the founding fathers of synthetic biology, they were all engineers, not a single biologist, okay? What you may welcome or not, I don't know, but the idea is that sometimes biology is too important to leave it just in the hands of biologists, and then you have to bring other people to really take care of the important matters there, matter for discussion. Okay, so what are the consequences of considering these metaphors and this idea of applying the same logic that engineers use for constructing their things to biological systems? Okay, so you say, well, what is a typical biological, a typical engineer object? Well, for instance, a radio, and this is not a modern radio, it's an ancient radio. You have here a transistor, well, the transistor, you have these bulbs here, you have a speaker, you have a box, you have like screws here. I mean, these are typical engineer object, something that has been engineered by men. Okay, so if we apply this metaphor to living systems, why don't we think on the fact that bacteria, I mean, in the previous slide, say, well, this is a radio that has many parts, and I can list a list of parts that are involved in the construction of this radio. So maybe in a biological system we have something similar. So we have a large number of molecules, of components and so on, but it's a limited number, so it's not an infinite number. So by the same token that an engineer can take a radio and start disclosing the radio in many independent parts, then a synthetic biology can take a cell and make a list of parts. So say, well, what do we have in a cell? Well, I'm sorry that the slide is a bit blurry, but I think that the concept is very clear. You have a cell here, and then you have genes, you have DNA, you have some, components of the membrane, you have promoters, you have, terminators. I mean, you have, in a cell, you have a large number of structural components that are not different of what a radio may have in terms of components. So one very important concept in synthetic biology, this is something that we will spend nearly one whole lecture on that, is the concept of parts. What is a biological part? You know that the concept of part comes, for instance, car engineering. You take the car to the garage, and then you need to have a list of parts, or please replace this part or whatever. So in biological systems, or in synthetic biology, a part is the minimal biological object that delivers a completely defined function. Okay? So in this case, for instance, we have a repressor, a very single repressor. And you know that repressors, again, we'll return to that in detail in subsequent classes, their only function, their only mission is to bind to DNA and repress transcription. So it's a very single repression here, okay? But we have many other types of parts. We can have not only repressors, we can have promoters, we can have terminators, we can have reporters, we can have small RNAs, we can have, biosensors. I mean, you can have a tremendously long catalog of parts. But remember, the number of parts that form a cell is not infinite, it's finite. So it has, is limited, it's a very high number, but it's still a limited number. And therefore, it's like a car or a radio. You can have a list of different parts and different components. So what happens when an engineer takes parts and combine them together? Then they produce a device. And what happens when various devices are rationally put together? Then you have a system, okay? So this is something that is what is called an abstraction hierarchy that is typical of engineering. When you want to build something, you start first making a kind of abstraction of what you need to deliver that particular activity in an engineer object. So you have to start with parts, then the parts have to be combined to produce devices, and eventually devices have to be combined to produce systems. Let's see whether we find equivalence of these things in biological systems. I'm going to put to the side just to change a little bit. Okay, so we had a part before. A part was a repressor. What happens when we combine two repressors? For instance, we have a construct of DNA. We have this repressor that is called the lac repressor, and then this repressor has been engineered through standard genetic engineering techniques to drive expression of a second repressor. Okay, so we have here a situation in which we have combined 1, 2, 3, 4 different parts to produce a device. What parts are here? Well, we have a piece of DNA that is called a lac operator. That is the segment of DNA that is recognized by the lac repressor. Then we have, after that, we have a ribosomal binding site that is necessary for translation. And then after that, we have the gene of another repressor, is the lambda repressor. At the end, we have a segment of DNA that is a terminator. Well, this is a super simple device, okay? You will see in many examples later that you can make devices with many different components. So by making this combination, you generate a device that happens to be an inverter. Inverter means that the input and the output are related by a transfer function that is inverse. So that means when you have a lot of this Ci here, when you have very little of lac Y here, then lac Y is unable to repress this promoter here, and then you have a lot of Ci here, that is the repressor that is here, okay? So you have this, this input and this output. Then you see that when lac I is little, you have a lot of this. But as you go on and increase the concentration of lac I, at some point, you start repressing this site here, this promoter, and therefore the amount of this product here starts coming down. So you have here an invert, say, relation that can be described rigorously mathematically. You have an inverter. So the amount of one input is exactly the opposite of the amount of the output, okay? So this is what engineers would call an inverter, okay? So an inverter is a device. It can be electronic or it can be any other, say, material, say, basis, where you have an input and you have an output, and both are related through an inverse function, okay? A lot of lac E means very little Cy, and vice versa. But now you can go on and say, well, let's see what can you do with an inverter. For instance, one thing that engineers can do with inverters is to put 3 inverters in a row and then to generate an oscillatory system. And, well, this is just an abstraction. So theory will tell you that if you are able to connect 3 inverters, one after the other, then you generate a system that will oscillate, okay? Because there's some time between the moment that you enter an input here and the moment that you have an output here, and then because of the delay, this is not an instant transmission, it has some delay. This delay will result in a oscillatory behavior. Now, can we do something similar with biological systems? Say, well, at the end, I want to have a system that oscillates, for instance. And then to do that, I need to have 3 inverters put in a row, one after the other. I say, well, this is what you need theoretically. Question for a biologist, can I have 3 inverters? Yes, and then to have 3 inverters, you need to find somewhere 3 devices that can be connected together to form this system here with an oscillatory behavior. And then the device is composed by parts. So you have to find the parts that will originate, will produce this inverter here. And then at the end, you can encode everything in DNA. So once you have the DNA in place, then here's the DNA. Then here's a set of DNA binding proteins that is recognized and you need DNA site. And then you have a set of these proteins here so that each of them sends and receives a signal that at the end will turn into a oscillatory behavior. So there's this concept here, it's called POPs. I will return to that at some point. That means polymerase per second. So polymerase per second is the synthetic biology equivalent of electric current. Don't be concerned about that. We will explain that in detail. But basically, these arrows here in electric devices is electricity. In genetic devices is POPs, is RNA polymerase that will transcribe each of the members of this device there. Is this theory? Okay. So because of this consideration that I told you, here comes the first paper that is considered the foundational paper of synthetic biology. And is this paper that was published by someone called Mike Elovitz with this other here in year 2000. So they were, I will discuss briefly, this is one of the papers I want you to read in detail because I think they are very, very important, very foundational. And well, in this case, this idea of having 3 inverters simultaneously is developed with considerable, say, detail. Okay, so what is this paper about? So as I told you, an engineer can construct an oscillatory system by putting 3 inverters together, and the inverters can be brought about with electronic devices. So what these people did was to ask the question, well, can we do the similar, can we produce a similar behavior on the basis of using not an electrical component, not an electronic component, but because of using just biological components? So what they thought that, well, we need, to have this oscillatory system with 3 inverters, and the way to generate inverters in biology is by using repressors. So you have repressor 1 that will repress expression of repressor 2, and then repressor 3 here. So what repressors, how do you implement that in biological terms? Okay, you can make a plasmid, a construct, in which you have, as you have a plasmid, you need to enter a part in the plasmid that is an antibiotic resistance, because eventually it has to go to a cell. You need a promoter that drives expression of one of the first repressors. Then you need a second promoter that will act on the second repressor, and then another promoter that acts on the third repressor. But look at this. This is the whole trick of this is the kind of genial idea that these people have. What if I make this promoter of the second repressor to be repressed by the protein that is produced by the previous promoter here? So you have promoter 1, promoter 2, promoter 3, and the result of each of the promoter actions will be the production of a repressor that will repress the subsequent promoter here. Okay? So the prediction, the theoretical prediction, is that if you are able to do that, you will get an oscillatory behavior. Because if at the end these repressors control, I will show you in a moment, a plasmid that has a reporter system, then the reporter system will follow an oscillatory behavior. This will be obviously an ideal situation, okay? A prediction, a simulation. So how does it work in reality in this paper that I just showed you? Okay, so what these people did was the following. They used 3 repressors that are known in the E. coli literature. The tetracycline repressor, or TET-R, the LAC-I repressor, that is the LAC repressor, and the lambda repressor. And they made a construct in a plasmid in which they had the tetracycline repressor, TET-R, controlling expression of the next one, repressor, LAC-I, and LAC-I controlling expression of the next one, that was the lambda. And everything was in a plasmid with an original replication coming from another plasmid. Okay, so, and then how can you read, what would be the readout of all these, of all this construct? So what they did was to put in the same cells another plasmid that had a fusion between a repressor that was controlled by the tetracycline repressor here, okay? So you have a reporter here, and this is driven transcription from a promoter that is controlled by TET-R. That is one of the repressors that you have here. So at the end of the day, what happens is that TET-R will inhibit production of GFP. GFP is a fluorescent protein, okay? So what they did was to put these two plasmids in a cell and wonder what happened, you know? And what happened is this. You go to the paper, I think that in the paper there's a link to a video, so you can even see these cells, say, blinking. So they focus in one cell, and then in one cell they saw that depending on the time, and look at the periods of the time, is a considerable number of minutes here, 60, 100, 40, blah, blah, blah. And then if you focus in one of the cells, and this is, I think in the paper you have a color video, so it's better than this one. But if you focus in this cell, you see that this cell now is off. Now this is on. Now this is off. Now this is on. Now this is more or less off. This is on. This is off. This is on. Okay? So by using this technology that enables you to capture the output of one cell in a microscope and monitor what is going on, what they obtained was something like this, okay? Okay, well, you may say, well, I mean, isn't this amazing? You know, this is the first time in history that by using a rational engineering approach, we could make bacteria to behave in a fashion that they would never, ever behave themselves, just by applying engineering principles. Because the engineering principle, you have these three devices that you put together with the expectation of having an oscillator. And well, you may say, well, this is an oscillator so-so, right? I mean, because in the previous slide, you know, the idea is that you have these wonderful kind of oscillations, and here you have a kind of, I don't know, oscillations, but they are not very impressive. However, this is a big principle, a big start, because, you know, there's no way evolution could have invented this. However, by using this engineering approach, then these guys, these folks, were able to really demonstrate that you can implement in a biological system something completely coming from an engineering concept, and you can implement it not with electronic devices, not with, say, screws, not with lights, but with pure biological systems. I predict, I may be wrong, that these two guys, in particular Mike Elovitz, sooner or later will get a Nobel Prize, okay? Because this was such a smart way of proposing that you can engineer biological systems by using engineering principles, that I think that they deserve all this credit. It's interesting that sometimes some of these developments now we take it for granted. You know, we say, well, this is, I mean, so what, you know? But by the time, it was a super big revolution, conceptual revolution. Now, there's another issue here, and I will stop at that point, the first part of the talk today, and is also a foundational paper, very important paper. They appear nearly simultaneously, and it was the construction of a genetic toggle switch. Again, this is something that has been invented by engineers a long time ago. Well, I don't think we use them much these days for lights, but your toggle switch is this device, electronic device, that is either on or off. You cannot have an intermediate step. Either you are all on, or you are all off, but you cannot be in between, right? So, can we implement something similar in bacteria? Okay, so these people here that came from Arizona laboratory, run by this gentleman called Jim or James Collins, then figure out the following. What if we, again, apply to a biological system something that comes from engineering? And is that we have two mutually, say, interacting little networks that we can engineer in terms of DNA sequence in such a way that we have two inducers, and then one of the inducers will take the system completely in one direction in the other inducer will take the system completely in the other direction, but in such a way that there cannot be anything in between. Okay? And this is the basis of the construct. If you think about it, you use this inducer, for instance. Well, let's suppose that I use inducer number 1. The inducer number 1 will de-repress this repressor here, and then that means that this promoter will be activated, and then the repressor 2 will be on, and therefore the target promoter that might be out there it will be inhibited here. Okay? And then the contrary in the other side here. Okay? So by doing this genetic device, then they showed that indeed the system can only have 2 possibilities. Either you are entirely on or you are entirely off. So this is seen better here. This is a system for monitoring gene expression that is cell cytometry. So cell cytometry is a procedure that will tell you whether one gene in particular is being expressed in a population and what is the level of expression. So in this case, with one of the IPTG induction here, then you see that either you are completely in one of the sites, or when you induce with the other inducer, in this case it's not a chemical inducer, it's a temperature, then you have this other situation. So you see, you jump from here to here, from here to here, but you never find anything in between. So it's like here, you go from all to nothing. There's nothing in between here. So this is one case in which, this is something that hardly ever happens in nature, So in nature, normally you have inductions that you add an inducer, and then it goes little by little growing. In this case, what you have is a situation in which you jump from one stage to the other. Well, this is not this is not correct in the sense that I said that this never happens in nature. It happens sometimes, and I have the time in one of the classes I will mention that. But this is an entirely engineered situation, rationally engineered with a model behind, and in a fashion that indicates that again you can implement a behavior that is not natural in a recombinant bacterium by doing these tricks. Okay, so I will stop at this point to make a little break. But I think that in this part of the talk, I just wanted to bring you to your attention that the essence of synthetic biology is just to apply engineering to biological systems for the sake of understanding how they work, but also for the sake of programming them to do something that they normally don't do. And I think that this whole idea of the repressilator and the whole idea of the toggle switch, not without reason, were good examples on how you can really apply these rational systems or these rational, say, frames to program bacteria to do something that is not natural. Well, that's a bit the story, but I keep on I leave you with this slide for now, and you see that this is not perfect. I have to say that now there are better versions. Keep in mind that this is 20 years ago or 23 years ago, a long time ago. Probably many of you were not born by that time. And subsequent improvements, now you have better oscillations and you have behaviors that are way more complex, but this is a seminal paper that is cited in every, say, case, in every course on synthetic biology as one of the, instances where you can really see the power of rational engineering for making this type of things. In the second part of my talk today, I will show you other more flashy examples on something similar in which you can really even convert E. coli into a photographic material. Well, I will show you that later. But for now, we'll make a little break here. How are we doing with the time? It's 3:30. Now, questions before we make a little break. Okay, both. Yeah, well, both. Okay. So in many cases, you start with existing parts. And the existing parts have a context. You have a gene, you have a promoter, you have a Schendel-Garn, you have a terminator, this and that. But the part itself is the part. However, in many cases, the behavior of the part can be improved in 2 ways. One of them is to have a different context, to have better promoter, to have a different Schendel-Garn, to have a terminator, and then the activity of the part improves a lot. In the other cases, what you can do is to have variants of the parts through mutagenesis. Okay, so imagine that you have an enzyme that converts product A into product B, and product B is very valuable. And the basis of that transformation is an enzyme that has a given activity. So how can you improve that situation? So you can either have a better promoter, have a better expression of the gene, or you can have a variant of the protein itself that will do better. Okay, so that would be one possibility. But there's another possibility that is becoming more and more, say, interesting in the last few weeks, I would say, because this field. Well, one thing I have to tell you that the field is moving so quickly that one can hardly even keep track of the literature. So every week there's something completely amazing in the field of synthetic biology. One of the very, very latest developments in the biology is to have completely artificial proteins. Okay, so proteins that are not found in nature now can be, in some cases, designed by artificial intelligence, machine learning. We will return to that at some point as well. That produce activities or produce enzymes that do not exist simply, or they were not evolved. So the answer to your question is that you can do both. I mean, you can mostly so far use parts that are already there, even though we can manipulate them a little bit, or to enter into the equation parts that are completely artificial. And that's something that is growingly possible. More questions, more comments? How do you like it? I mean, you think that, how do you react to the fact that biological systems can be engineered, not just metaphorical, but engineered in earnest, to go deep into the engineer biological systems and then program cells and everything? Well, this is what I told you so far is nothing compared to what I will tell you later and also in the subsequent steps, right? Because once you have the power to engineer biological systems, I mean, there's no limit to what you can do. One of the things that I will tell you, and in fact it's a project in which I'm partially involved, is the following. I mean, you can think the craziest things that you can imagine, but still they may have some basis. So you have trees, right? So the shape of the trees is the result of a biological program, right? I mean, not all trees are the same. They have different branches, they have different leaves, they have different architectures. But at the end, the shape of a tree is determined genetically. So that means that the program that originates that particular architecture is encoded in DNA, right? So now, what if we were able to really decode the program that originates the architecture of a tree, right? Then, for instance, one thing that we can do would be to program a tree to develop as a house, for instance, okay? You say, well, this is crazy. No, it's not crazy. I mean, we're not there yet, but why not? I mean, there's no reason why you can do that, because DNA is omnipotent. I mean, the moment you know how to write a program in DNA, there's nothing you cannot do. I mean, you can make absolutely amazing things. And one of the, say, possibilities of the future is to have this organic architecture in which the components, the parts that we use for construction of buildings are not, cement and these things, but using things that are produced by trees themselves. Obviously, they have to be, you may call them manipulated or programmed or you name it. And, but why not? I mean, there's no reason why you cannot do that. And also, by the same token, we can produce medicaments, we can produce new materials. I mean, there's no limit to the possibilities that biotechnology has now by applying synthetic biology to many, many different issues here. And if you go through the IGEM projects, and at some point we'll make an exercise on the IGEM, say, platform, then you will see the craziest projects you can imagine implemented with the tools that I just mentioned, okay? And therefore, it's up to your imagination to find applications to this type of approaches. More questions, more comments? No? Okay.